Friends, this lecture is about artificial intelligence in plant breeding. This comes under modern era of plant breeding, which was introduced after 1990s and going on till now. As you know, genetics and plant breeding are divided into four different eras. First is pre-Mendelian era, then Mendelian era, post-Mendelian and modern era. In pre-Mendelian era, this started around 10,000 years ago, where conventional breeding, domestication, artificial pollination, hybridization, and phenotypic selection were the major tasks. In Mendelian era, which started in the year 1900, before as well as after years it has covered, where hybridization, progeny testing, pure line selection, pedigree method, bulk population method, and backcrossing were the major techniques. In post-Mendelian era, this is around 1980, this is also called as Green Revolution era. Here, marker-assisted selection, genetic mapping, and genomic selection are the important topics given importance. In the modern era, genome editing, high throughput phenotyping, G2P prediction, predictive modeling techniques, then ML and DN, and integration of AI are the major techniques. So, in this lecture, we are exclusively covering artificial intelligence. As you know, this is a cutting edge technology. Cutting edge means more advanced and innovative ideas which may improve different facets of the any technology. Okay, so rapid advances in genomics, phenomics, and molecular biology are accelerating crop breeding into the era of artificial intelligence, addressing the formidable challenge of managing the data deluge. Here, data deluge means huge data which is unmanageable, which is crucial for transitioning from disparate data sets to an integrated data infrastructure in AI enabled plant breeding. Means through artificial intelligence, the huge data generated in plant breeding procedure can be managed. Although AI has revolutionized various aspects of crop breeding, such as phenomics, variant calling models, gene discovery, genomic selection, gene editing, there is a precise need to pressing need to synthesize these components into an integrated breeding techniques for future crop development. Before getting into the topic, we should understand some of the terms. The first one is artificial intelligence. This is a suite of technique including ML, DL, and natural language processes to stimulate human intelligence process by machines. The process include learning, reasoning, and self-correction. In learning means acquisition of information and rules for using the information. Reasoning means using rules to reach approximate or definite conclusions, and self-correction. Convolutional neural network, CNN. This is a type of DL algorithm. DL means deep learning algorithm designed specially for processing and analyzing visual data like images and videos. It mimics the structure and functioning of human visual cortex by using layers of neurons to extract features from input images. Next term is data islands. This term field usually refers to isolated and disconnected sets of data that are not integrated and interconnected. A major application of AI enabled plant breeding is to access diverse and comprehensive data sets to train robust and generalized models and predict the plant performance to select ideal plant types. Deep learning. It's an AI component process to train artificial neural network or ANN with multiple layers to learn a representation of the data. Each layer of the neural network process of the data in hierarchical manner by extracting increasingly abstract and complex features from the input to anatomically discover patterns and features from raw data without the need of explicit programming. Next is deep neural network. This ANN architecture characterized by multiple layers between the input and output layers. Then gene editing, genomic selection. These are the common techniques as well as old techniques. Coming to machine learning, it's a subset component of AI that can develop algorithms and statistical models that enable computers to perform tasks without explicit instructions. Machine learning uses an algorithm to learn from patterns in a large amount of labeled data, and once trained, prediction or decisions based on the learning can be made in response to new and unseen data. The last one is variant calling. This is a process of identifying variations in a genomic sequence by comparing it to a reference genome or another set of genomic data. Major type of the variants are SNPs or SNPs, small insertions or deletions, that is indels, and large structural variations. This variant calling is a nice approach of artificial intelligence which solves major of the problems in plant breeding. Coming to the topic, artificial intelligence is renowned for its prowess in big data analysis and pattern recognition and is revolutionizing numerous scientific domains, including breeding. Here, various sets of breeding include data collection, unlocking genetic diversity within gene banks, and bridging the genotype phenotype gap to facilitate crop breeding. In the next slide, I'll explain it with a figure. Here, just I am reading this for your knowledge. This will enable the development of crop cultivars tailored to the projected future environments. AI tools also hold promise for refining crop traits by improving the precision of gene editing system and predicting the potential effects of gene variants on plant phenotypes. Leveraging AI enabled precision breeding can augment the efficiency of breeding programs and holds promise for optimizing cropping systems at the grassroots level. This entails identifying optimal intercropping and interrotation models to enhance agriculture sustainability and productivity. Compared to traditional methods, AI tool can extract more useful and less biased insights from high throughput sequencing and imaging data. Large language model, that is LLM based ChatGTP, serves as a potent chatbot that can generate intelligent text and image based neutral natural language input. This tool has been instrumental in posing thought provoking question relevant to plant science, filling gaps left by plant experts in projects like 100 important questions facing plant science. This is important figure where four facets of AI in breeding are clearly explained. In first one, panel selection, where from gene bank so much genes or depositions are found. Here, AI enabled methods for predicting germplasm's performance in target environment, which was enhanced by spread breeding and breeding materials, where large pooling are taken for generating the breeding material, which are in smarter way. The second one is biological big data. Here, marker assisted selection, sequencing, multi-omics like phenomics, genomics, all omics are used here. Test in multi-environmental means genotypic, environmental, and multi-location traits are studied here. And again, phenomics is done with the help of drones, sensors, where different softwares are used like ResNet 50, GoogleNet, YOLO OV3, like that. Then in phenotype prediction, casual variant, then gene mining with which genetic network with biological interpretation is done. From there, genome editing can be done with AlphaFold 2, then smart breeding platforms with the selected lines, and finally designing new variety by excellent haplotyping methods, then with identifying the novel loci and ideal combination of the chromosomes as well as chromosomal segments. AI in germplasm utilization. We'll see one by one. Here, high throughput advances in remote sensing and plant omics have provided scientists with large multidimensional data sets to work with when addressing the problems. Use of machine learning algorithms in pre-breeding strategies, regional selection, and adaptive marker assisted selection can boost genetic diversity and speed up the development of cultivars that are climate resilient. These algorithms can also maintain and restore the dynamic processes that result in genetic variability, which is necessary for plant adaptation to changing climate. Datasets can serve as foundation for the targeted selection and optimal testing of accessions in specific environments using AI driven predictive genomics. Second is in phenotyping. Phenotyping platforms equipped with advanced image sensors have potential to revolutionize large scale phenotyping of various plant traits and environmental conditions. These platforms may utilize either stationary or mobile sensors. Towers and other fixed platforms are commonly used to monitor growth phases. Then, high resolutionary cameras can be used here, which takes the pixel densities even nearly 1 mm, means that much minute pictures or minute changes can also be monitored and taken into account with AI based precision phenotyping. And high resolution UAV photography can also be possible by this. See in the picture plant phenomics here in indoor, then handheld tower, rail based platforms, phenocart, and UAV. These different techniques are used with different sensors and with different trait studies. Then different AI tools like ML and DL architectural studies. Then training and application of AI of model for spike identification, means with the help of different software developed specifically for plant breeding, this can be studied. Next is AI enabled predictions to explain genomic data. Numerous studies have demonstrated the potential application of AI in interpreting biochemical data to advance the understanding of plant stress biology. Example, AI has been efficiently used to forecast genomic crossovers in maternal and paternal maize plants, thus aiding in the identification of genomic regions with elevated mutation rates. Based on the DNA methylation pattern of maize plants growing under stress, researchers identified AI approach to identify and characterize genomic area, thereby differentiating between functional genes and pseudogenes. Likewise, AI algorithms examine the expression patterns of important genes to predict gene promoters and cis-regulatory elements in Arabidopsis thaliana and maize plants. Then for multi-omic big data in plant breeding. At biochemical level, omics encompasses diverse molecular data sources like genomics, epigenomics, protein DNA interactions, transcriptomics, proteomics, and metabolomics. In past decade, vast amount of omic data have been generated, inundating the web with the transcriptomic, then genomic, proteomic, and metabolomic information, where now biology has evolved into an information intensive science because of the massive data sets generated by high throughput submolecular biological experiments across various domains, including genomics, transcriptomics, proteomics, and metabolomics. In bioinformatics, the inventory of components at the genome, transcriptome, and many facet level is gradually becoming comprehensive and invaluable to researchers. ML is frequently used to assess vast datasets because their size, complexity, and requirement for combined interpretation. Even within each omic technology, several profiling techniques have also matured in addition to RNA sequencing for genome wide gene expression, chromatin immunoprecipitation with deep sequencing, that is with chip sequence, DNA affinity purification sequence, that is DAP sequence, and assay for transposase accessible chromatin sequencing, that is ATAC sequencing. AI enabled bridging the genotype phenotype gap. The development of cultivar with high yield potential that are resilient to climate change relies heavily on genetic improvements through the identification of novel alleles and genomic assisted selection in breeding. Missing heritability refers to the instance where all the genetic variants cannot completely account for the heritability to complex traits and is a major bottleneck in genetic studies. In such cases, integrating transposons and epigenomic mutations like DNA methylation can offer more comprehensive information to explain the genetic foundation of phenotypes. There have been massive advances in the identification and classification of transposons in plants using DL, including impactor 2 and tesorter. In addition to AI enabled gene identification to bridge the genotype phenotype gap, or genome phenome gap, AI has promising applications in functional genomics of the genes identified to construct genomic intelligent design. Example, GANs or generative adversarial networks were used to optimize and enhance genomic functions via synthetic promoter design in E. coli. Means here, by using epigenetic mutations and DNA methylation patterns at different locations and interpreting the results, means by collaborating the different data, genotyping phenotyping gap are overcome by AI techniques. Then functional genomics and gene mining using AI. Several ML methodologies have emerged to prioritize genes relevant to agronomic traits like leveraging gene functions, exploring protein interactions, incorporating gene annotations and sequence variation. An intriguing avenue for further exploitation lies in integrating evolutionary insights into ML models, and exciting frontier where ML plays a crucial role is in single cell RNA sequencing. Then the next is finding excellent alleles and causal variants from omic data. Coming to applications of AI in gene editing. Here, the recent technology is CRISPR Cas, and AI has been used to characterize structural information and optimize protein functionality, basically in genome editing. This could revolutionize public sector plant breeding program like those under the one CGIAR, means as many research institutes are under CGIAR, just like that many technologies will come under AI to improvise the breeding. Data driven decentralized breeding program like these can predict crop performance better than conventional genomic selection. AI enabled breeding platforms are accelerating the breeding process by deploying advanced computing and analyzing algorithms. The promise of AI in gene discovery and allele mining is well defined. The different further challenges may be to stimulate and characterize the diversity of gene banks for predicted environments, to introduce novel traits into cultivars without linkage drag. Then the second challenge is construct the capacity and training human resources to efficiently utilizing computational power for AI enabled predictive breeding. Then offer multidisciplinary AI training to breeding teams engaged in designing future crop cultivars. And the fourth one is to produce a unified plant breeding cyber infrastructure instead of data islands. The utilization of AI has also been demonstrated great promise in predicting the best cropping pattern and system by integrating big data obtained from physical sensors, UAV platforms, and Internet of Things, that is IoT devices under diverse genotype into environment into management practices. This would complement the AI based genetic innovations to obtain the required rate of genetic gain to meet the food and nutrition challenges in the next decade. This is all about different facets where AI is enabling in the plant breeding process, means this is making the conventional breeding into smart breeding as well as speed breeding with different softwares as well as different tools, and that is a step towards to meet the food and nutritional challenges for the next decade. This is all about AI in plant breeding. Coming to AI and robots in plant breeding. AI assisted breeding system will play a significant role in theoretical study, evolution, selection, breeding procedure, development, and field management. These equipped robots will interact with all processes involved in data collection, storage, analyze, sharing, utilization, significantly upgrading breeding information system. Then it also benefits from historical experience and relevant knowledge produced and accelerated in breeding programs. Breeding systems driven by big data and AI will have great capacity for design and prediction through model simulation and optimization. There are different robots like AlphaFold 2 AI robot algorithm significantly outperformed other teams in protein folding context at the critical assessment of structural prediction. This is all about AI. Thank you. If you have any doubt, you can keep it under comment section. Thank you once again.